right, so we're talking about cellular respiration. So if you've had lab this week already, you probably slightly touched on it a little bit at this point. But now we're going to dive in a little bit deeper. All right, there's the key concepts in case you would like to know them. But let's begin with just an overview of metabolism and cellular respiration. All right, so these chemical reactions are going to occur in these metabolic pathways. All right, they are going to be connected reactions together. There's going to be a ton of different reactions that make up metabolism. All right, but they're basically in two different pathways that we can separate them out. All right, catabolytic pathways, which is where we are breaking down a cellular component. So maybe we're taking a macromolecule, making it into a monomer, perhaps. So think about like the breakdown of carbohydrates, like starches or glycogen. That's pretty common. And in catabolic pathways, we are releasing energy, right? So they are exergonic, meaning they are spontaneous. We are releasing energy. Now, the other pathway that metabolism can fall into is the anabolic pathway. All right, this is where we're synthesizing cellular components. All right, so we're making larger molecules. Now, this is endergonic. All right, so oftentimes it's coupled with an exergonic reaction in order for it to happen spontaneously. Now, depending on which pathway we're talking about here, each step is going to be coordinated by a very specific enzyme. All right, so if we look at just this one example up here, all right, in each step we're adding on a phosphate group to this glucose molecule. All right, this enzyme is very specific to step 1, right? And in step 2 we have a different enzyme that is very specific to step 2. And same with the third enzyme, all right? Different enzyme, very specific to this step, all right? So there's a lot of enzymes in these metabolic pathways here, and they're all very specific, and they're all pretty much different. All right, so let's look at catabolic reactions a little bit more in depth. All right, so we're breaking down a reactant. We're breaking down a larger molecule. And there are a couple of reasons for us to do that. All right, so one reason might be so we can recycle different building blocks. All right, a good example of that is in proteins. So sometimes proteins become denatured. Maybe they're improperly folded. Maybe we just don't need that protein anymore. All right, we can break down that protein into its components, break those peptide bonds, get individual amino acids, and recycle those amino acids back into a larger molecule. All right, so we can break down, so we can recycle components. We also use catabolic reactions to use energy to drive endergonic reactions. All right, so this is very common. We're going to be talking about this quite a bit. So when we break a covalent bond, right, for example, right here, we have a pyruvate molecule with a phosphate group attached. All right, this just happens to be a very unstable bond. All right, so when this bond breaks, this covalent bond breaks, it's releasing energy. And that energy is going to be stored in some sort of an intermediate molecule. Now, in this case, it's ATP. Right? ATP is a pretty common one. NADH is another fairly common one as well. All right, so we're storing that energy in these intermediates. All right, now with these reactions here, they're typically going to be redox reactions. All right, so we're either going to remove electrons or add electrons to another molecule. So oxidation is the removal of electrons. It's called oxidation because oftentimes oxygen is involved in some shape or format. And then we have these reduction reactions. All right, so it's the addition of electrons to another molecule. Right? And it's called reduction because we're adding on a negatively charged electron to another molecule. We're reducing the charge of that molecule. All right, so if we look here, we have this A molecule with an electron on it, and we have this B molecule, right? Oh, and I clicked the button there. We are going to oxidize the A molecule, so we're going to get rid of that electron and move that electron from the A molecule to the B molecule. Right? That B molecule is reduced because it has a lower charge now because we added on that negative electron. All right, so this can happen in a couple of different ways depending on how we want to look at an equation. So if we look at the top equation here, this is just kind of the general equation for cellular respiration. If we look at glucose here, glucose is losing hydrogen atoms. All right, so we can talk about electrons. We can also talk about hydrogen atoms in terms of redox reactions. Losing hydrogen atoms, so it's becoming oxidized. All right, so in this oxygen molecule here, all right, we're actually gaining some hydrogen atoms to make water, so it's becoming reduced. All right, so that's pretty straightforward, easy to understand. We can also talk about these reactions in terms of the individual atom within a molecule. And this is where we have to think back to electronegativities. All right, so if we look down here, this is the same reaction, all right, but the electrons are drawn out. All right, so if we look at glucose here, all right, we see this C to H bond here has 2 electrons. It's a polar or a non-polar covalent bond, so those electrons are shared equally. All right, but once these electrons get moved, all right, so now we have carbon still, all right, but the oxygens are further away, or the electrons are further away from the carbon. All right, so we can say that this carbon atom has been oxidized. All right, so you can also think about this in terms of electronegativity. All right, so we know oxygen is highly electronegative, highly more electronegative. No, what am I trying to say? Has a higher electronegativity. How about that? So it has a higher electronegativity, so it's kind of hoarding those electrons towards it. All right. So a couple things to remember when we're talking about these redox reactions. All right, so molecules with many C to H bonds have a lot of potential energy because they are non-polar covalent, right? Because they are sharing those electrons equally. Now, oxidized molecules, they're often going to lose a proton, which is this hydrogen atom, right? Hydrogen only has 1 proton and 1 electron. We lose the electron, it becomes just a proton, so that H+. Right? They're often going to lose a proton and an electron and increase the number of C to O bonds. Right? The C to O bonds, they're more electronegativity, high electronegativity, which means they're going to have a lower potential energy. All right? So through these reactions, the potential energy is going to decrease. All right, any questions so far? It's a lot of information. All right, so let's talk about NADH here. So we talked about how that electrons that are removed by oxidation are going to be moved into some sort of energy intermediate, such as ATP or NADH. All right, now NADH, all right, is let's see, NAD plus is nicotinamide. Oh gosh, I practiced this one because it's such a mouthful. Nicotinamide adenine dinucleotide. All right, I'm just going to say NAD+. All right, now once it receives the hydrogen and electrons, all right, it becomes NADH. And this molecule is super useful. You can kind of think of it as like an electron carrier. All right, it easily gets reduced and easily gets oxidized. So it can be really easy to carry along those extra electrons in these redox reactions. All right, so it can release a lot of energy when it's oxidized to make ATP, and it can donate electrons very easily during synthesis reactions to help give them energy. So NADH, very, very powerful. All right, just because kind of like an overview of what the molecule itself looks like and what is actually happening. All right, so this is our NAD plus molecule. If we add on 2 electrons and a hydrogen, all right, or a proton here, all right, we're going to reduce it. All right, and basically we're adding on that hydrogen atom up to the top here and changing the number of double bonds that are present, as well as taking away that positive charge from the nitrogen. All right, so this is actually what's occurring when it's being reduced and when it's being oxidized. It all has to do with these 2 electrons and 1 proton. All right, so just as a quick note on the anabolic reactions, all right, we often call them biosynthetic reactions because we are synthesizing biomolecules. And we're just making smaller molecules into larger molecules, but they require some sort of energy input. All right, that energy input can come from NADH or ATP, and they can help to drive those reactions. All right, so we talked about redox reactions. We've talked about NADH. All right, we've talked a little bit about the overview of metabolism. All right, but with everything in our body here, there's going to be regulation involved. All right, so we can regulate our metabolism in three kind of different ways here. All right, it can be genetic, it can be through cell signaling, it can be through biochemical regulation. All right, so if we talk about turning genes on or off, so this is where we're regulating things at a genetic level here. We can basically turn off the synthesis of a certain protein if we need it or if we don't need it. So a good example of this is enzymes. All right, we can turn enzyme synthesis on or off as needed. So say we have an enzyme for a very specific sugar molecule. All right, if the sugar molecule is present, then the genes will be turned on and we will synthesize that enzyme. If that sugar molecule is not present, we're not going to turn on the genes. We're not going to synthesize those molecules. We're going to conserve that energy. All right, so that's one way we can regulate it. Another way to regulate these are cell signaling regulations. All right, so this involves the protein kinases. All right, so these are enzymes that covalently attach a phosphate group to a target protein. And a good example of this is epinephrine. All right, so kind of that adrenaline protein or adrenaline molecule here. So epinephrine, that's a signal to your muscle cells to start phosphorylating enzymes in order to activate them, to activate carbohydrate metabolism. All right, so if we're activating metabolism through this pathway, we're basically saying, give me more energy, I need more energy. And that helps with that flight or fight response that we often hear of. All right, so that's a good example of cell signaling regulation. And then the last one is one that we're mostly going to be focused on for the rest of the class here, is the biochemical regulation. All right, this is basically feedback inhibition. All right, the product of a pathway inhibits the early steps to prevent the overaccumulation of a product. And this is where inhibitors on enzymes are going to start to become very important. So let's take a look at that. All right, so we need to maintain appropriate level of substances within our body. All right, and there's a whole range of substances that we have to keep at these appropriate levels in order to maintain that homeostasis. All right, so this feedback inhibition kind of prevents the formation of too much product during a metabolic pathway. So let's look at the example down here. All right, so we have our initial enzyme here with our initial substrate. All right, we create intermediate 1, goes on to another different enzyme that creates intermediate 2, goes on to yet another different enzyme, and then creates the final product. Now, as we get more and more of this final product, right, this final product is going to start to act as an inhibitor on that initial enzyme, right, inhibiting the production of more of this product so we don't overaccumulate. All right, now notice this one is in the allosteric site. It's not binding directly to the active site. And it's causing a conformational change, right? Shape equals function. And because the shape has changed, the enzyme can no longer function. All right, homeostasis is a big one. All right, so I want you to kind of work with each other here, break apart, take a break from listening to me talk at you, and use the internet to find examples of substances or processes that need to be regulated to maintain homeostasis within our body. All right, so take a few minutes to do that. I expect to hear a lot of examples on this one.
Okay, wait. You said enzymes.
Yeah, like the production of enzymes, the synthesis of them. Yeah.
Okay, but not
Yeah, feedback inhibition. Yep.
Okay.
So I mean, turning genes on and off, that can happen with other types of molecules as well.
Okay.
Oh yeah, no problem. Oh yeah. All right, it's getting really quiet, but I can't tell if it's because you're done answering the question or because it's a Friday. So, anybody need more time? All right, I feel you. I see you guys dragging a little bit. Is that part of the semester? And it's a Friday. I'm also dragging a little bit. But who has an answer for me? And what needs to be regulated in our body to maintain homeostasis? Temperature. Yeah. And how might we regulate that?
Sweat.
Sweat. Yes. What's another example?
Blood sugar.
Blood sugar. Yes.
Toxins.
Toxins. Yes.
Blood pressure.
Blood pressure.
Heart rate.
Heart rate.
CO2 levels.
CO2 levels. Anything else that hasn't been mentioned?
Against virus
Perspiration, hormones, viruses, pretty much anything that happens in our body is going to be regulated to some degree. All right, so maintaining homeostasis is huge. All right, and because metabolism is so many different chemical reactions, all right, there's a lot of regulating that goes on in our metabolic pathways. All right, so let's jump in to cellular respiration. And we're only going to talk about half of it today. All right, so cellular respiration, we need to obtain energy from organic molecules, primarily to make our energy intermediates such as ATP and NADH. All right, now we're mostly going to be talking about aerobic respiration, the use of oxygen. Now in a couple weeks, we'll start to talk about fermentation and anaerobic respiration, but for the next couple of lectures, we're going to focus on oxygen. All right, oxygen is consumed, CO2 is released. All right, so respiration also involves us breathing, right? Breathing in the oxygen, releasing the excess carbon dioxide. Now we can use a variety of organic molecules to go through cellular respiration. One of the most common ones is glucose, and so that's what we are going to be focusing on for our class. All right, so let's look at specifically glucose metabolism. But these four metabolic pathways will be present in all the other molecule pathways as well. So we have glycolysis, we have the breakdown of pyruvate, we have the citric acid cycle, also known as the Krebs cycle in some textbooks, and then oxidative phosphorylation, which is also known as the electron transport cycle in other textbooks. All right? So we're going to talk about it as four pathways. I know you've probably learned in the past as it being three pathways, right? Sometimes this breakdown of pyruvate is either clumped with glycolysis or clumped with the citric acid cycle. We are taking it out as its own separate pathway. All right, so the generalized equation is right down the bottom there. There's a lot of steps to get to this final equation. All right, so brief overview. Glycolysis here. All right, as we go through, pay attention to how many carbon atoms are in each molecule. All right, that's a good way to kind of walk yourself through cellular respiration. So we're going to start with glucose and glycolysis. All right, 6 carbon molecule. All right, now in glycolysis, we are breaking that down into 2 pyruvate molecules that are 3 carbons each. Now this is happening in the cytosol. All right, we are not in the mitochondria at this point. This is happening outside in the intercellular fluid, outside of the matrix of the mitochondria. All right, so it's a big distinction. Oftentimes that's forgotten about when we start thinking about the details of cellular respiration. Make sure you bear in mind where each of these steps are happening. All right, so after we created 2 pyruvate, all right, in glycolysis, we yield a little bit of ATP, we yield a little bit of NADH, all right, but the main product is pyruvate, and that's going to go into the breakdown of pyruvate stage. All right, so it's exactly as it sounds. It's going to be broken down into an acetyl group and CO2. All right, CO2 as a byproduct. All right, so now we go to the citric acid cycle. All right, and let's see. So a little bit of NADH is produced in this step as well, but the citric acid cycle is the next big one, also known as the Krebs cycle. It's called the citric acid cycle because we are essentially binding this acetyl group to an organic molecule. That organic molecule is typically citrate, so hence where the name comes from. And we're going to oxidize that molecule to make CO2 and ATP. right? Again, a little bit of ATP. CO2 is our byproduct. And again, we're producing that NADH and another molecule, FADH2, right? It acts very similar to NADH, so I'm not going to go too in depth on that molecule. Finally, we have oxidative phosphorylation, all right, or the electron transport chain as it's commonly known as, where molecules are going to be oxidized to create large amounts of ATP. Right? It is in this step that oxygen is consumed. We're going to focus a little bit more in depth on glycolysis. All right, so glycolysis and the breakdown of pyruvate is what we'll be looking at today. Glycolysis is step one. All right, so glycolysis, that can occur with or without oxygen. All right, and it's actually pretty interesting because it's thought that this process evolved very early, right, when the Earth's atmosphere did not have plentiful oxygen like it does now. So, right, this is a really old step in cellular respiration. And there's basically 10 steps in glycolysis that we're going to break down into 3 phases. And what's really cool is that these 10 steps and these 3 phases are nearly identical in all living organisms. All right, so this is a highly preserved pathway throughout evolution. So let's start looking at each of these steps. And as you're studying glycolysis and all the cellular respiration steps here, what I'm showing you here is what I expect you to know. All right, so for example, there's 10 steps. We're not going to go over every single 10 step. What I'm showing you here, for example, energy investment is steps 1 through 3. All right, now in each of these steps, a different molecule is formed, but I only expect you to know the fructose 1,6-biphosphate. All right, so that should help your studying. Don't get too bogged down by the details here. All right, so in energy investment, we are using 2 ATP molecules to adhere these phosphate groups onto our glucose molecule, and that turns that glucose molecule into fructose 1,6-biphosphate. All right, now by adding on those phosphate groups, we're essentially increasing that energy of the molecule so it can help drive other exergonic reactions down the line. All right, cleavage phase is the next phase. And this is where we're taking our 6-carbon molecule and breaking it down to 2 3-carbon molecules. And that molecule is glyceraldehyde 3-phosphate. Let's see if I wanted to say anything else about that one. No? All right. I'm trying to keep it as simple as I can when we're talking about glycolysis. All right, so we're breaking it down into 2 3-carbon molecules. And then this last step, this is where we're going to get most of the production of ATP in glycolysis. In fact, all the production of ATP in glycolysis. This is step 6 through 10. All right, this is where the 2 glyceraldehyde 3-phosphate molecules are broken down into 2 pyruvate molecules. All right, in this step, we're producing 2 NADHs, which will be used later on when we talk about oxidative phosphorylation. We're also producing 4 ATP. All right, now when we're talking about glycolysis, we typically say there is a net gain of 2 ATP, right? But we're producing 4. Now we say there's a gain of only 2 because we are immediately using these 2 back up in that first step. All right, so we're using 2, we're gaining 4, so it's a net gain of only 2. All right, so I put this slide on here just for your own review. It's just everything we just talked about without the pictures, all in one slide. Net yield of 2 ATP. That's a big takeaway here. All right, so we know that living organisms maintain homeostasis, and this part of cellular metabolism is no different. All right, we have to maintain a relatively constant level of ATP in the cytosol. Remember where this is happening not in mitochondria, this is happening outside of mitochondria in the cytosol. And that's going to be controlled by that feedback inhibition that we talked about. Specifically using this enzyme, phosphofructokinase. All right, again, another mouthful. And that's going to catalyze that 3rd step in glycolysis, which I highlighted right below. All right, now at high levels of ATP, that's going to inhibit the function of this enzyme, and it's going to cause that feedback inhibition and kind of stop glycolysis from happening for the time being. Right? So basically the cell is telling itself, I have enough energy right now. I can wait to do glycolysis for a bit. Right? So it's inhibiting itself. Now, one thing that your book mentioned, which I think is really cool, cancer cells have unusually high levels of glycolysis. And we can actually see this through different imaging technology that we have. So many diseases are actually associated with alterations in carbohydrate metabolism. Right? And this is in cancer cells called the Warburg effect, where cancer cells are preferentially using glycolysis while decreasing oxidative phosphorylation. Right? So they're mainly using glycolysis to get their energy. Now this is thought to happen because tumor cells have low oxygen levels. Right? So if they have low oxygen levels, they can't really go through the whole step of cellular metabolism or cellular respiration. So it's using glycolysis. Right? So it's overexpressing those glycolic genes in response. And we can see this on different types of scans. Right? So this is a PET scan, which is a positron emission tomography. Right? I don't expect you to know the name of it there, but that's what it stands for. And basically what we have done is this individual has, I don't know if they have them eat something or inject it into them, but they are taking in this radioactive glucose that this scan will pick up in its imagery and show us where in our body we have high concentrations of glucose. And that's going to indicate high glucose using cells, which typically indicates high levels of glycolysis. All right? Glucose is only really used in glycolysis because after glycolysis, it's changed its form to pyruvate. Right? And in this image, we can see that they're mostly in these tumorous areas. Right? And it's pretty interesting that this happens in about 80% of all types of cancers. Right, so a cool little side note about glycolysis here. All right, so far we've been talking about glycolysis, which happens in the cytosol, outside of the mitochondria. All right, next we're going to be talking about inside of the mitochondria, and specifically the mitochondrial matrix. All right, so remember mitochondria has two membranes. Inside of that double membrane system is the matrix. But that matrix is outside of this system of cristae, all right? These flattened membrane sacs, essentially, all right? So it's all those spaces in between. This is where the breakdown of pyruvate is going to happen. So this is step 2. It's going to be transported into the mitochondrial matrix. Now it is a relatively larger molecule, so it uses these channels and these symporters to get through both membranes. And once it gets into the membrane, it's going to be broken down by the enzyme pyruvate dehydrogenase. All right, so essentially a molecule of CO2 is going to be removed, and it's going to leave an acetyl group. That acetyl group is going to attach to a molecule of CoA, which is a short lingo for saying coenzyme A, and that creates acetyl-CoA. Right? It also creates NADH in the process. One NADH for each pyruvate. Right? Now that acetyl-CoA will end up going on into the Krebs cycle, all right, or the citric acid cycle, and it's going to be broken down even further. All right, we're going to get out of here early today, which is good for a Friday. But before we do, I have a group question for you all. All right, I want you to work with a partner and explain to each other the 3 phases of glycolysis. All right, what are the products formed in each phase, and what happens in this step? Or where does this step occur?
Oh no, I didn't. That's it.
All right, does anyone need more time to answer this question? All right, so first of all, where does this occur in the cell? In the cytosol, yes. All right, so what's the first phase of glycolysis? Energy investment phase, yes. And what molecule is created at the end of this phase? Yes, I hear some rumors here. So the fructose 1,6 biphosphate. And do we use ATP in this stage? How many molecules? 2. All right. So the next stage, who has that one for me? Cleavage, yes. So what is the end result, molecule that results from this cleavage stage? Glyceraldehyde 3-phosphate, yes, and 2 molecules of those. All right, is any ATP used or made at this step? No, all right. So the third step, energy liberation. Yes, and what molecule is created at this step? Yes, so pyruvate, yes, that's created, 2 molecules of pyruvate. We're also creating an NADH molecule. But how many ATPs are we producing? 4. So this whole step of glycolysis, what is the net yield of ATP? 2. All right. All right, and I had additional slides here just to uncover it as we talked, which I forgot to do. So that's all I had for this lecture. I anticipated there being more questions. Yes. So enjoy your extra time. You all look dead. But there is an assignment. All right, before you leave, there is an assignment. Due Monday before class. All right? It's online. It's super easy. It's having you watch a video, read an article, and answer. I think there's 5 questions to it. So on Monday, be prepared to talk about the Nobel Conference, and I will have your exams back.
On Monday?
On Monday, yes.
Oh, okay.
It's like a pre-Nobel.
Okay, I was gonna say, I was like, wait, the Nobel isn't.
Yes.